This is the mini lecture for membrane transport. Cell membranes separate the contents of cells, which we can consider living, from the outside or non-living environment. Therefore, cells, they're sometimes known as the boundary of life. We have a picture here of some eggs, and the shell is not the cell membrane, but there is a cell membrane inside of the shell. It's important to remember that eggs can go bad over a long period of time, and that this is because they continue to exchange gases like carbon dioxide and oxygen with the external environment. Just a note that if you're prepping for the end of the world, you can preserve your eggs in mineral oil, which will slow the exchange and allow them to last for a much longer time. Aside from the end of the world, the major component of a cell membrane, they're those phospholipids we discussed in a previous module. Remember, these look like a balloon with two strings, and then the head is polar, and the tails are nonpolar. Their orientation in what's called a lipid bilayer, it has them facing tail to tail, with the polar heads exposed to the water both inside and outside. And this creates this like hydrophobic region of the bilayer across which many polar molecules cannot cross. However, gases like oxygen and carbon dioxide, they're nonpolar, and they're freely to cross in and out of this membrane. The lipid bilayer it's the primary component of a cell membrane, but there's other items that are like inserted into or attached to the lipid bilayer. Cholesterol is added to the lipid bilayer to give it some rigidity. The cell membrane it's got this sugary coating on the outside of it. It's made of glycolipids and glycoproteins exposed to what's called the extracellular fluid, the outside the cell. And finally, there's various proteins that are embedded into the cell membrane or attached to it. Cell membrane proteins, they have various functions. Proteins embedded in a cell membrane can function as enzymes, catalyzing reactions. Attachment proteins can secure the cell to the extracellular matrix, basically just a scaffolding of proteins. Receptor proteins are important on cells like neurons and muscle cells that receive signals from other neurons. Receptor proteins usually connect with hormones, things like epinephrine. In fact, when you have an adrenaline rush, many organs in your body, like your trachea and your heart, they all respond to epinephrine or adrenaline flowing through your bloodstream because they have these receptor proteins. One of the most important ones that we'll discuss time and time again in this course are transport proteins, which are in the cell membrane that help with facilitated diffusion or active transport. Junction proteins can bind one cell to another cell. The cells in your skin have very, very tight junctions to prevent the entry of microbes and damaging chemicals. And finally, there were those glycoproteins that were attached to the outside of the cell, and they're capable of connecting with other cells for recognition. In fact, these are the types of proteins that are used by your immune system to recognize your own cells as well as foreign cells. Diffusion is the movement from an area of high concentration to an area of low concentration. Check this out. I made this like 12 years ago. Woo! You want to see it again? I can't show it to you again. So this movement that you saw, it's a passive movement with no energy required. You're familiar with this if I were to put a droplet of ink into a glass of water. Over time, you would see that the ink molecules disperse or diffuse out to areas of lower concentration, moving from high to low. Over time, this will result in the ink molecules being evenly dispersed throughout the water, creating a homogeneous mixture. So this, what we just described, we described diffusion, a movement, but what we had there was a concentration gradient. And concentration gradients determine the movement of molecules. They determine which way the molecule is going to move. In this picture here, we have a concentration gradient with lots of solute molecules, the green bubbles, on the left, and not many solute molecules on the right. So the solute is going to move from the left to the right, or from the area of high concentration to the area of low concentration until there is a homogeneous mixture. The molecules will not move from right to left or from low to high concentration because energy would be required for that movement, and that's like against the concentration gradient. Tonicity. It's the concentration of solutes. For our purposes in this class, a solute is going to be a gas or a solid within a solvent. And for our purposes in this class, a solvent is going to be a water. So I have drinks here that are ordered from left to right, and they're ordered in increasing solute concentration or increasing tonicity. If we start with water, pure water, and we add carbon dioxide, we get seltzer. If we add salt to our seltzer, we get club soda. And if we add quinine and a whole bunch of other stuff, we can finally get tonic water. We can say that these solutions are ordered from left to right, from hypotonic to hypertonic. These words are relative, such as seltzer is hypotonic to club soda, and that club soda is hypertonic to water. You picking up what I'm laying down there? Think about it. Osmosis is the movement of water only through a selectively permeable membrane. Therefore, osmosis is the movement of water into and out of cells through the lipid bilayer, but also through proteins embedded in the cell membrane. Water will move to an area that is hypertonic, an area with a lot of salt, to make it a homogeneous mixture, to basically flood it out or dilute it. I like this cartoon down here because it reminds you that osmosis is the movement of water only. I don't know why this cartoon has to be Abe Lincoln, but I guess that's because it really hurts being called a fool by Abe Lincoln. I'll give you a moment to read it. I can hum during that moment. I don't remember the rest of the Jeopardy song. All right, moving on. We want to learn about osmosis in order to determine if cells will shrivel or burst in certain types of environments. The rule to remember is that water follows salt. And what this means is that water will always flow to an area of hypertonicity in order to try and flood the salty area. Many diuretics make you pee, removing water from your body, but what they actually do is they remove salt, and the salt thus draws more water along with it. These cells here on the left are submerged in a hypertonic environment, and therefore water will want to leave these cells and flood the hypertonic environment. These red blood cells will eventually shrivel, called crenation, and this plant cell here will do something called plasmolysis. These cells here on the right are submerged in a hypotonic environment, and therefore water will be drawn into these cells, which would have a hypertonic environment, and these red blood cells would ultimately burst. The plant cell here would become turgid or pressurized with water with that nice full vacuole. Many students have a common misconception that when there's a homogeneous mixture between the outside and the inside of a cell, that movement ceases, and this is untrue. When there's an isotonic environment, water continues to be exchanged freely between the outside and the inside environments. This is actually what cells strive for. And this is homeostasis. Facilitated diffusion is a movement down a concentration gradient from high to low, but requiring a protein to cross the plasma membrane. Water and other polar molecules are not able to cross the lipid bilayer because they get repelled by that hydrophobic region created by the nonpolar tails of the phospholipids. Therefore, a transport protein, like this big purple one in the picture on the right, it acts like a bridge spanning the lipid bilayer and covering the hydrophobic region. Now, the polar molecules are free to move down their concentration gradient. Water crosses into and out of cell membranes using a protein called an aquaporin. When you drink a diuretic like coffee or alcohol, aquaporins are removed from the urine-generating tubules in your kidney, and you're incapable of conserving water. I don't know about you, but this is commonly known as breaking the seal, and it's an undeniable urge to urinate. Also, the urine will be incredibly dilute and flooded with water that you're unable to conserve. Active transport, it's essentially the opposite of all of these passive movements: diffusion, facilitated diffusion, and osmosis. Active transport is the movement against the grain or against the concentration gradient, moving a molecule from low to high concentration. This requires energy in the form of ATP. I sometimes feel as though my life is like active transport, moving against the concentration gradient and requiring massive amounts of energy from me. Anybody else? Anyway, I have a picture here of an interesting little protein called the sodium-potassium pump here on the left. And you can see it uses ATP to maintain a sodium and a potassium gradients in your cells. You can see that the pump is moving sodium from its area of low concentration to its area of high concentration. It will also move potassium from its area of low to its area of high concentration.